course.
Helps a lot.
All right.
So I went through CP and uh BB, like so chem phys and biochem. Um, it's pretty interesting in chemistry and physics. Like it's genuinely, I'd say, all calculation errors and
Got
just
it.
like question misinterpretations, passage misinterpretations, a lot of just like a lot of stuff that I just could get correctly on the second try on my blind review.
Yeah.
So it was just it was really honestly infuriating. Super infuriating. Like, yeah. I also do think that the Jag Weston exam was a little more calculation heavy, in my opinion.
Yeah.
But anyways, that's that.
Yeah.
But on the biochem, there's like a decent amount of content stuff. Um, also I feel like there were some low yield things that just like I don't know. I like cis and trans Golgi faces. Like I never that's like the lowest yield thing ever. Like they were testing us on whether we know a difference between like because the cis side faces like toward the roughy or whatever. But yeah, so, like a couple little things that nuanced things that I didn't get. But
Hmm.
what I'm realizing, it's just like as I said in the I don't want to waste too much time discussing this because we already did through email, but like it's really annoying that I like know most of the stuff, but it's just like just I am sometimes overthinking and sometimes underthinking in terms of what the question is asking. And I'm just misinterpreting what the question is asking
Let's
sometimes.
let's pick a
Yeah.
passage
Yep.
like from like CP and let's go over it and I'm gonna have you take the reins and you're gonna kind of go like read it and kind of like tell me what you're thinking as you're reading it.
Yeah.
Hmm.
Second, I'm opening the
Hmm. So, and it's good that we know that a good amount are the calculation errors, so we have something to, work on.
yeah.
And the Sisson trans Golgi, that's I wouldn't say it's low yield because you want to know that if you're taking a protein and you want it to become part of the cell membrane,
Hmm.
it's gonna come out of the trans side of the Golgi.
Hmm.
And if it's something, that is going to be within a protein that's within the cell, it can come out of like the cis phase. But yeah,
Okay.
vesicles, right, would be going that way. Yeah.
All right, here. So this is actually a Biochem passage, but I just remember doing this and like I was just like I know the content, or at least I feel like I do, but I was just like misinterpreting, I guess, just what was going on. Um, it's about the nervous system.
Okay.
And here, so we'll go through it and I'll tell you what I'm thinking each step.
Um, let's see. Okay.
And I got every question wrong in this passage. So
Okay.
um taurine, a non-proteinogenic amino acid, is an agonist of GABA and glycine receptors in the nervous system. So agonist, um, it's something, it's I mean it's the opposite of antagonist. I always think of antagonist as something that inhibits. So an agonist like increases, I guess, like um it increases the activity of whatever. It's agonizing.
Yep, yep. Because like an antagonist is like the bad guy in like a movie or
Yeah.
show. So an antagonist will work against it, and an agonist would work like for it. So
Yep.
have taurine, which is in a lot of energy drinks, by the way,
hmm
non-proteinogenic amino acid. What do you think that could mean?
It does not create, it does not create proteins. It's an amino acid that doesn't, it's not part of like polypeptides.
Good, good. And so they said it's an agonist of GABA and glycine receptors in the nervous system. Um, Anything about that?
Yeah. So when I see that, agonists, you're increasing GABA and glycine activity. GABA and glycine are inhibitory um,
Yep.
neurotransmitters, right? And GABA, I know, is inhibitory for the brain. Glycine is for the spinal cord.
Okay.
So,
Good, good, good. All right. Good.
okay. Local taurine application to synapses activates chloride conductance, similar to GABA and glycine. So here is where I like kind of like was just like, I don't really know what this means, but I'm gonna keep going. I ended up getting a question that was like on chloride, I think. So, that was obviously not
Hmm.
the best thing. So yeah, I wasn't fully sure. Yeah, like I know Cl minus, if Cl minus enters the presynaptic neuron, I know that it would lead to hyperpolarization because it becomes more negative. I know that, but I'm not sure if it's the same thing.
So yeah, maybe we can say taurine proportional to GABA, glycine, and then maybe inversely proportional to like, let's say, nervous system activation. And also proportional to chloride conductance. Because yeah, as you said, the for a synaptic for an action potential to happen, you have like, you'll have like a sodium influx, which will make it depolarized and more less negative, more positive. So if GABA and glycine are inhibitory, they're gonna cause it to, instead of becoming less negative, become more negative. So that could be the chloride aspect of it. Um, Good. Okay.
Okay, besides fast synaptic inhibition, tonic inhibition is described in thalamic neurons. Um, I always forget this, but I think, tonic, it's like when it's what, like continuous kind of like,
Mhm.
but I know there's also something where it's like acetylcholine is continuously binding and something that's twitching continuously, but I don't know if that's tonic. Like, I always confuse that with like tonic inhibition.
Yeah, so tonic inhibition is gonna, yeah, reduce the excitability of certain neurotransmitters. Um, or sorry, certain types of neurons. So yeah, yeah, you have the right idea. So yeah, we have some type of tonic inhibition in thalamic neurons. Okay.
Um, Tonic inhibition results from continuous activation of extrasynaptic GABA receptors in response to low concentrations of ambient GABA.
Okay. So we know that this tonic inhibition happens when you continuously activate these GABA receptors in response to low concentrations of GABA. Okay.
Mhm. Scientists investigated that. Hold on. I want to understand first of all what I just read. Um, Wait, so
Yeah.
extrasynergic GABA receptors are like in the synapse? Like, are those just the receptors that are like on the postsynaptic, like in the cleft and the postsynaptic neuron?
Um, let's see. Extrasynaptic GABA receptors. Um, I guess that could mean receptors that are outside of the synapse, so
Mhm.
elsewhere on the neuron, perhaps.
All right. Scientists investigate the excitability of neurons with taurine using current clamp recordings from the mouse ventral basal thalamic neurons. Okay. Excitability. Okay. The current clamp method records membrane potential following an electric stimulus. Recording was continued with a subsequent application of Gabazine. Okay. Strychnine, a glycine receptor antagonist, was present in the media and throughout the experiment. So glycine receptor agonist, it's basically inhibiting glycine. So less inhibitory response. Right. And that was present in the media.
Okay.
Right.
Results are shown in figure one. Figure one, representative current clamp trace, firing evoked by a point 1 nanoamp current in VB neuron to facilitate measurements. Membrane potentials were depolarized to about -60 millivolts. Okay, so we're being told that this current clamp method records the membrane potential after an electric stimulus, and that the recording was continued with application of Gabazine, whatever that is. And then we talk about strychnine, which is an antagonist of glycine, so gonna function very differently from taurine and GABA and glycine, of course. Um, and if we take a look at the figure here, we can see, I guess, in the control that we have, these spikes that are happening relatively frequently. When we have the taurine, we see that the spikes are happening less with a lower frequency, right? Because we have a smaller number of these, you know, peaks.
Mm-hmm.
And that should make sense because we did say that taurine is inversely proportional to the nervous system activation, right?
Mm-hmm.
Now, Gabazine plus 50 micromolar of taurine seems to raise the number of these, you know, peaks significantly, right?
Yeah.
So Gabazine is likely to behave like strychnine in that it's going to produce more nervous system activation, right?
Mm-hmm. Yeah. Yeah.
So, okay, that's consistent with, what we know from the passage that taurine is going to act like. And now we know that gabazine seems to be working in an opposite way from taurine.
Okay. Um, Apart from neuromodulation, extracellular taurine plays a neuroprotective function and counters glutamate-induced elevation of intracellular calcium concentration.
Okay. So we can say it can also be inversely proportional to um, glutamate, which is that excitatory one, right? And we know that glutamate causes a higher amount of intracellular calcium. So we can say, inversely proportional to calcium ion intracellular.
Mhm.
So, okay, good. Continue.
Taurine protects neurons through two mechanisms. Okay. Firstly, taurine indirectly regulates the activity of the ER and the plasma reticular Ca2 plus ATPase. So it regulates activity of an ATPase, which so
Mhm.
I guess it uses ATP to pump Ca2 plus.
Yeah, so when they say regulate, we don't know yet if it causes something to happen more or happen less, but we do know that it is going to affect the endoplasmic reticular calcium ATPase.
Mhm.
Well, I guess if we know that it counters the elevation of intracellular calcium, then perhaps it could mean that,
Yeah.
like, if this ATPase breaks down calcium, then it can perhaps upregulate that. If it doesn't, if it makes calcium or whatever, it can be inversely proportional to that. But yeah, we don't know yet. Uh, good. Continue.
Second, the taurine transport mediates loss of taurine from cells accompanied by sodium loss. Um,
Hmm.
mediates loss of taurine. Okay, I mean like the taurine just yeah, it allows taurine to leave and
Hmm.
sodium also leaves with it.
Yeah.
Is that a form of symport or like you know what I'm referring to?
Yep. Yep. So yeah, I don't know. I don't think it's part of anything like that, like any particular receptor made for taurine. Um, so yeah, I'm not sure yet.
Consequently, less sodium is available for calcium entry via the Na plus Ca2 plus exchanger. Okay. That exchanger, it can operate bidirectionally, allowing Na plus to flow down its gradient across the plasma membrane.
Okay. So yeah, so taurine can change the activity of the calcium 2 plus ATPase, and um it can also affect the loss of taurine from the cells, and that's accompanied by sodium loss. And I guess that can be considered like a symport if they're both leaving. And then we said consequently less sodium is available for calcium entry via the sodium calcium exchanger. So if you have the taurine cause a loss of sodium, then you're going to have less of this MCX exchanger functioning, right?
Well, yeah,
Mhm.
but less sodium, less NCX, yeah.
Mhm. Got it. Okay. So good. Now let's try these questions. So let's try this one.
So how do you, like you were saying, how do you then read slow while still having time for everything?
Um, well, we read this slowly and we were able to write out these proportionalities, which means that for this question, for example, we already have that answer, right? Um, Meaning the passage mentioned Gabazine, didn't tell us what it does, but in figure one here, we see that, well, first, let's say control over here, and let's just say we have 1, 2, 3, 4, 5, 6, 7, 8 of these peaks, right? And when we add taurine, we have 1, 2, 3, for 5 of these peaks, right?
Mhm.
And that should make sense because we know from the passage that they said that taurine acts like GABA, and GABA is an inhibitor, which means that you should have less action potentials, right? Less of these depolarization peaks,
Okay.
right? So it makes sense. It should make sense that in the control, if we have 8 of these peaks, once we add taurine, we should have fewer than 8, right?
Yeah.
Okay.
So
And now for this one, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So the Gabazine is giving you a higher number of peaks than even the control. So we know that the Gabazine is going to be working against taurine,
right.
right?
Yeah, it's excited. It's gonna lead to excitatory
Uh-huh.
responses.
Yeah. So basically, so we already when we read this, because there's like, let's say there's 2 approaches, right? There's the approach of you reading quickly and not looking at the figures and just going through, and then when you see this, it says figure 1, and then you go back to figure 1. That's fine. But I think that, like, on the test, right? When you see a figure or a graph or something like that, I think you should give it 15 seconds to get the gist of what it's saying. So that was when we looked at this and we saw, hey, Gabazine. So I should say 2 things. So we started off talking about all this stuff, and then they said something with application of Gabazine, right? They didn't tell us anything else about it, right? So when we saw this figure, that's where we can figure out what Gabazine will do, right?
Yeah.
And so if you see that figure and you just spend 15 seconds to get, the main, the gist of what it's trying to show you, it seems like what it's showing us is that yes, compared to control, taurine reduces the number of these spikes, which makes sense because we know that it's going to behave in an inhibitory fashion. Now, when we have Gabazine plus same amount of taurine that reduced the amount of the spikes, we see a higher amount of spikes, higher than even the control, right?
Yeah. So I mean, I get it. I get it. Like this question.
So
Um,
just tell me, like, when you were answering this, what were you thinking? Like, if you walk me through your mindset, like what you were thinking.
I think I was just rushing and I like didn't even stop and think about um because I was thinking more of like trying to remember what exactly I was thinking, but
So like what you saw based on figure 1, right?
yeah.
Go to figure 1 and then look at that.
Yeah, I did. And for
And
some reason I thought it was in like inhibitory. Like it was like I thought the opposite of what I think now. I just don't remember why.
uh-huh. Got
I
it.
was just rushing.
Yep, yep. So, okay, okay. Um, so now we know what to do, that, we go back. So they say a figure, we go back to the figure in case we didn't, figure it out the first time that we looked at it. Um, and when you look at it, you can see that it's behaving in a way that's the opposite of the way that taurine behaves. And if we look at these choices, right, that can also be because, yeah, all right, we're going to have multiple ways to figure out the answer, right? So let's say that you weren't able to right away know that this is behaving the way that it does. Glycine receptor agonist, GABA receptor agonist. We know that GABA and glycine are both inhibitory, but we don't know how to differentiate the two, right?
Yeah.
Because they're both, like, this could be both. Well, theoretically, like, depending on what they show us, like, there's no way for us to know if it's going to be like doing something to glycine or doing something to GABA, right?
Yeah, yeah.
So, and we know that, so if it was a glycine agonist, we should see more of an inhibitory effect and less of a smaller number of spikes, right?
Yeah.
And we know for the GABA agonist, it should also do that, right?
Hmm.
Because also they told us that taurine is an agonist of GABA and glycine. So if taurine lowers the number of these spikes, taurine should behave as either A or B, right? Or both.
Yeah.
Now, let's look at what about glutamate receptor antagonists. So glutamate, is an excitatory one, and if it's a glutamate antagonist, it should be inhibitory, right?
Exactly.
So these are inhibitory, and this is inhibitory. This, since we know that the GABA agonist is inhibitory, a GABA antagonist must be stimulatory. That's not really a word, but
Yeah, I get it.
so yeah. So yeah. So I think
It's honestly a little scary
yeah.
because I I mean it's kind of like maybe the id and like the ego and superego thing. Like I know what I should be doing, but
Yeah.
there are certain times where I like realize I was a little behind on like the first passage and
Yeah.
like my like unconscious just takes over and just
Yeah.
I just start like speeding through it and I'm like just trying to get like every question in, you know? Like
Yeah.
I don't know how to explain it, but it's just like I know deep down that I should be reading slowly
Mhm.
and the same thing was happening on Cars too, where it's like later on, for example, I was just
Yeah.
like, I know I should be mapping and for every paragraph, write what I need to,
Yeah.
add each question, structure, main idea, but I'm just like rushing to it.
Yeah. 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 No, that's it's definitely normal. Um, and yeah, we're gonna basically get to the bottom of this by having you, I guess, basically tell me what your thoughts are. And I know, like it's hard to remember, especially like when you're rushing and stuff. But okay, I think as we continue, we'll get a better idea. But from here so far, it's just that we see in the figure that taurine is doing one thing and gabazine seems like it's doing the opposite thing. And and even if we didn't know that, we can tell that, or we can do that the strategy of which choice is different.
Yeah.
Okay.
Okay.
So, all right. Now, when you read this, if you remember, tell me what you remember.
Yeah. Um, so basically I saw chloride, I started panicking, I'm
Mhm.
pretty sure, because I remember reading about chloride here,
Mhm.
and just skipping over it. I was just like, okay, hopefully this doesn't show up. They didn't mention chloride anywhere else, really. So like,
They wrote
um,
it. I thought, oh, yeah, sorry, go on.
yeah. So yeah, I mean, I saw it and I
Mhm.
I'm pretty sure. I mean, it was literally just like I was just trying to get through this as soon as I can, as fast as I can. And I think for some reason, because I saw like the exchanger,
Yeah,
I thought it was secondary active transport.
got it. So I think what happened there was the calcium and chloride maybe you mixed up because they start with C.
That's who maybe.
So, yeah. So yeah, if it's any of the active transport choices, that would be more so with calcium because they mentioned, ATPase, right?
Mhm.
Whereas the chloride one they mentioned up there, which is they said something that happens when you have taurine or GABA or glycine. So we know that the chloride thing is going to be something that's going to be happening with either taurine, glycine, or GABA. So if they're asking about chloride ions um, initiated by GABA, we should have chloride conductance. And what would that mean? that would mean that we're doing something with chloride, um, particularly, um, let's see, they said tonic inhibition, excitability, chloride conductance. Okay, so maybe we can think of in what I guess happens in terms of chloride influx in a neuron that we know that that could cause um do you think okay this is a better question. Do you think chloride influx would make, let's say, a neuron um depolarized or like repolarized?
Yeah, I mean, as I said this, I said before when we were reading, when chlorine Cl minus enters into the presynaptic neuron,
Mhm.
it repolarizes. So it
Okay.
makes it more negative.
Yes, yes. Okay. So yeah, that could be a way to maybe remember, like when you read chloride ion, if you remember that it's negatively charged, it can help you differentiate that from the calcium one, which starts with the C, so it can be confusing, especially if you're doing things quickly. But calcium would be positive charge, right? So we know that if they're telling us that calcium's doing this ATPase thing, and that that can be the active transport, we can say that the chloride one wouldn't be any type of active transport because they never mentioned like that it's related to any type of ATPase.
Mhm.
But we can then think that because sodium influx, potassium efflux, all of that stuff happens through diffusion or facilitated diffusion, right? It depends on extracellular levels,
Okay.
right? Like if you have your neuron of sodium channel open,
Mhm.
it's just going down the concentration gradient because you have a higher sodium amount or concentration outside of the neuron and a smaller one inside,
Okay.
right? Because for the sodium influx, all we're doing is we're opening the sodium channel, right?
Mhm.
And when you open the sodium channel, you have the sodium influx because there's a higher amount of sodium outside than inside, and it just goes down like through diffusion, right?
Yeah.
And then we have a higher amount of potassium inside compared to the outside. So when you open a potassium channel, it just efflux, it gets out of the cell because it goes down its concentration gradient through diffusion, right?
Yep.
And so chloride is going to behave in a similar way, like either it's a higher amount outside compared to the inside, which will cause that influx, or vice versa. So we know that chloride ions, just like sodium ions and potassium ions, they're just going to go down their concentration gradients.
Mhm.
So that'll make it facilitated diffusion because facilitated diffusion is just diffusion, but it's going through like a transporter or like a channel.
Okay. Um, Simple diffusion is when it goes straight through the membrane.
Yeah, without anything helping it. Yeah.
Okay. Got it. Okay.
Okay. Now I would say add this to maybe like an Anki card. So basically, chloride concentration outside of the cell is a lot more than what is inside of the cell. So it's similar to sodium in that way.
Mm-hmm.
So remember that sodium has a higher concentration outside of the cell. Potassium has a higher concentration inside of the cell. Chloride has a higher concentration outside of the cell. So if you open up a sodium channel, it'll come in through facilitated diffusion. If you open up a potassium channel, it'll come out through facilitated diffusion. And if you open up a chloride channel, it'll come in through facilitated diffusion.
All right. Primary active transport is just like an ATPA, like, and it's, for example, like NAK ATPase. We have to use ATP for primary active transport, right?
Mm-hmm.
And secondary is like an antiport or symport, or what?
I think secondary active transport is when it couples it to something that would be like a facilitated diffusion type of thing. Um, like yeah, like in an antiport, yeah. So primary active transport uses needs energy and it gets that from breaking down ATP, whereas secondary active transport needs the energy and it gets it from being coupled to something that would go down its concentration gradient. So it can be coupled to something like facilitated diffusion, basically.
Got it. So it doesn't use ATP. It's just like, because like with symport, you're kind of like
Mhm.
pushing them two like together, and I
Yep.
guess that's coupling. And antiport pushing them against each other, kind of. It's coupling.
Yeah. So yeah, primary transport will break down ATP and get
Okay.
that way. But the secondary would be something that would be coupled to something that moves down its concentration gradient.
All right. What would be expected current climate traces with taurine if fra fraquinine was not introduced to the environment? Um, okay.
So what is strychnine like? What does it do?
Glycine receptor antagonist.
Okay. So what does glycine do to the number of action potentials?
So glycine lowers the amount of action potentials.
Good.
Antagonist, it would lead to more action potentials.
More action potential, you said, right?
Yeah,
So now, if we didn't add strychnine, what would happen?
more action potentials.
That's
I
it.
remember what was going through my head when I did this. I remember
Oh yeah.
because I thought they were trying to test us on, how like the action potential is all or nothing. For
Yep.
some reason, I thought that's what they're trying to test us for, and I put equal. But the thing is, um, this is already about what's happening after the action potential
Mhm.
like happens, right? So also C and D are different answers. I just realized. Like,
Uh, yeah. So D is saying same number as the control, and C is saying
just
same number. Well, actually, that would be in the control, they're still using the strychnine, right?
okay.
So I think C would be kind of like that. But yeah, no. Ultimately, here, what you're doing here is okay. Strychnine is an antagonist of glycine, and glycine causes less action potential. So if you didn't have strychnine, so if you had strychnine, you would have more action potentials. So if you didn't have it, you would have less, right?
Oh, wait. Oh, yeah, yeah. Glycine antagonist. Glycine is inhibitory. Glycine, less action potentials, but it's an antagonist to glycine.
Yeah, here, let me do this. So when we first did our proportionalities, we did taurine's proportional to GABA and glycine, and that's inversely proportional. I wrote first nervous system activity, but now I can write inversely proportional to the number of action potentials,
Hmm.
right? So, and then strychnine and gabazine are inversely proportional to the glycine receptor, which means it's going to be proportional to the number of nervous system action potentials, right?
Mhm.
So
Yeah.
for this one, we know that the strychnine causes more action potentials. If you didn't have the strychnine, you should have a reduced number of action potentials,
Yeah.
right?
Yep.
Okay.
Yeah, just another reason to go slower rather than faster. I was like
Yeah, yeah,
speeding through this. I think I just
yeah, yeah.
okay. Um, how would the calcium homeostasis shift if cultured neurons with taurine were subjected to a NAK ATPase inhibitor?
Okay. So they talked about the sodium-potassium ATPase, or they talked about a calcium ATPase, and they talked about um, sodium calcium exchanger, um, and how it can operate bidirectionally, allowing sodium to flow down its gradient. Okay, let's. So this is a different type of thing, right? The sodium potassium ATPase.
Yeah.
What would that do?
Um, well, the way I think about it is it's like it keeps the resting memory potential,
Yep, it keeps it
maintains it at -70.
because you have, yeah, because you have, you're pumping out three sodium ions for the two that you're pumping in, right? Or
Yeah,
yeah, sorry, three sodiums out, two potassium in.
yeah.
Okay, and we know that the taurine is going to, let's see, the cultured neurons with taurine were subjected to a sodium-potassium ATPase inhibitor. Well, they did mention some stuff about taurine being related to a few of these things. Let's see. Right, indirectly regulates the calcium ATPase, and that it also mediates a loss of taurine from cells, which comes with a sodium loss, and consequently less sodium is available for calcium entry via this NCX. Okay, so if we inhibited this sodium potassium ATPase, okay, let's think about what the connection between taurine and the sodium potassium ATPase can be.
Taurine. Okay, well, taurine is an agonist of GABA receptors, so that means there's increased inhibitory response. So something that's inhibitory, that's like repolarization.
Okay.
Um, I don't know. I feel like the ATP, NAK, ATP is that always kind of working, or
Uh-huh. And we know that from the passage that taurine like kind of goes in the same direction as the sodium, like the loss of taurine is accompanied by the loss of sodium. So if sodium causes, let's say, more loss, and this ATPase over here causes sodium to be lost as well, the effect of taurine would either increase or decrease, right? It wouldn't be C, and it wouldn't be how much calcium that you have, right?
Mhm.
So if you had that inhibitor that inhibits this ATPase, that should cause, well, the taurine effect to increase or to decrease. So let's see. The taurine counters glutamate-induced elevation of intracellular calcium, protects neurons through regulating the endoplasmic reticular calcium ATPase, and then the loss of taurine accompanied by sodium loss. So less sodium is available for calcium entry via this exchanger, and it can operate bidirectionally. Okay. If it operates bidirectionally, they're saying that it can allow sodium to flow down its concentration gradient across the plasma membrane. So there's more sodium on the outside. So this should cause sodium to leave the cell, right?
Okay.
Right.
Hmm.
If sodium flows down its concentration gradient, there's a higher amount of sodium. Let's see. Okay, so yeah, it seems like taurine goes in the same direction as the sodium. So if you inhibited the sodium-potassium ATPase, and that's something that would go against what taurine does, right?
I'm a little lost. Wait, so
Mm-hmm.
taurine, can you explain it?
Mm-hmm. Yeah. So, so okay. Let's see. Um, okay, there's a higher amount of sodium outside of the cell compared to the inside. And if sodium flows down its concentration gradient across the plasma membrane, it should move inside, right?
Yeah.
Okay. And if you inhibit it, and the sodium potassium ATPase is the thing that pumps the sodium outside against its concentration gradient, right?
Hmm.
So let's say this is in the neuron, sodium concentration is low and sodium concentration outside is high, right? And they're saying that this NCX allows the sodium to flow down its gradient across the plasma membrane. So that should be going from the outside to the inside of the neuron, right?
Uh-huh.
Okay. Now, the sodium potassium ATPase is the one that can pump in two potassiums and it pumps out three sodiums, right?
Hmm.
And this require like it's an ATPase because it goes against its concentration gradient, right?
Hmm.
Because you're using energy to pump three sodiums out, and you have a higher concentration of sodium outside, so it's going against its gradient, right?
Yeah.
Okay. So if you then inhibit this, it can help, I would say, like, okay, so let's say we inhibited this. It's not going to create that much of a high amount of sodium on the outside compared to the inside. And since taurine is the thing that lets the sodium just go from the outside to the inside, going down its concentration gradient, it should be functioning against that, right? Because the sodium potassium pump or sodium potassium pump is the thing that makes the outside sodium concentration larger than the inside sodium concentration, right?
Yeah, but okay, so you're stopping
Mm-hmm.
you're inhibiting the NAK. So there's gonna be more and more and more sodium buildup inside.
Uh-huh.
Um, NCX allows it to keep going inside.
Mm-hmm.
Um,
Yep.
and what I'm not understanding is how it relates to Ca2 plus, because like this, do you need sodium to go down to concentration gradient for Ca2 plus to also like enter, or are they exchanging kind of like an anti-Porter system, or are they coming in together like a sym-port system, or am I completely getting this wrong?
No, no, no. So they do say calcium entry via this exchanger. And it's weird because they're saying an exchanger, which to me implies that they're going in an opposite direction.
Yep.
But they did say that it can operate bidirectionally, uh, which allows it to flow down its gradient across the plasma membrane. So I think that would mean that if the sodium is coming inwards, the calcium should be coming outwards just from them saying that it's an exchanger.
Hmm.
So yeah, so I think that um if you inhibited the sodium potassium ATPase, which is the thing that makes sodium concentration outside the cell higher, and if taurine is the thing that can make the sodium go down its concentration gradient from high to low, like going into the cell, that if you inhibited the ATPase, the sodium potassium ATPase, it would reduce the ability of sodium to go from the outside to the inside. Let me write it like this. Sodium potassium ATPase is proportional to sodium concentration outside the cell, right?
Hmm.
Now, taurine, they're saying, is going to be in the same direction, either from them saying that if you have a loss of taurine, you have a loss of sodium. That can help us figure out that sodium and taurine are going in the same direction. But also here, sodium flows down its concentration gradient. So that would mean that taurine can help let sodium either go into the cell from this. Loss of taurine means sodium loss, so gain of taurine means sodium gain inside of the cell. Or this part that says the taurine lets the sodium flow down its concentration gradient. Both of this means that taurine is going in the same direction as the sodium. So taurine should be proportional to sodium concentration inside the cell, whereas the sodium ATPase, sodium potassium ATPase, is proportional to sodium outside of the cell. So they're operating in opposite directions. So if you inhibited the sodium potassium ATPase, you would have a higher amount um, of sodium on the inside. You know what? There, I just noticed this part about calcium. So maybe it depends on calcium, but let me see. Okay. Calcium entry, less sodium, calcium entry, right? So they're going to be in opposite directions. So, okay.
Yeah.
Back to this one.
What?
Come back to this one because I have to see it in the context of calcium. So now I'm thinking that it might be related to calcium because I didn't really think of it that way at first. But so far, this is the only question from this passage that could be a little weird. So yeah, I'll look into this one, but I at least for the rest of the questions makes complete sense the way that we did it, right?
Yeah.
Okay.
Um, let's see something. Oh, it's just annoying. It doesn't let me click reset the tab at the time.
I'll be right back. I'm just using the bathroom real quick.
Yeah.
Okay. So yeah, for that question, my instinct was this. It was that the sodium-potassium ATPase is what causes a higher amount of sodium to be outside of the cell. And the taurine, I would imagine, like causes more sodium influx, which is in line with what the sodium-potassium ATPase does. So if you inhibit it, my thoughts would be that you would reduce the effect of the taurine. But in light of seeing that they mentioned the calcium thing, I need to take another look at it. But all the other questions in that passage, the way that we went over it, should make sense, right?
Yeah.
Okay.
I think we could stop here just because it's like
Oh
an
yeah.
hour mark. I just yeah, I want to finish reviewing this exam and I'm seeing a couple buddies at 8. So yeah, I think we'll call it a day here.
Yeah.
But I think unless like we're going over a passage just that's very tough and like you want to take the reins, apart from that, I think I should always from now on forward read the passage because
Yep.
you're really helpful and this is why I always, I continue meeting with you. You're a fantastic tutor. I just think like me personally, right? When I during a session like and just listening to you talk and explain things, it feels good, right? But I'm not doing it actively, so then later on, like, you know, it's like watching a video basically. If I just listen to you, that's what I realized.
No, yeah, that's what I meant by saying, like, I even said that, like, yeah, we're gonna have you take the reins and you kind of walk me through what you're thinking, and all of that stuff. So, okay, awesome. Yep. So I'll see you tomorrow at 1. Okay.
Yes, I'll see you at 1.
Yeah.